A very good morning to all of you. Welcome you all in ISFCP dialogue series under the aegis of IQAC, IST, ISFCP, organized by ISF College of Pharmacy and Autonomous College, Goa, Punjab. Today's special guest and speaker, Professor Dr. Shamsher Singh, Head of Department of Pharmacology, ISF College of Pharmacy, and his title on Zebrafish as Emerging Animal Model. We welcome Dr. Shamsher in this ISFCP dialogue series, and you are requested to please proceed further. Please, sir.
Thank you, Dr. Mehenji. Thank you for your valuable information. So today in this particular lecture, I am going to discuss on the zebrafish, that how a zebrafish can become an emerging animal model, and what are the different areas in which the zebrafish can be a preferable species for the research, or it can be acting as a research tool, as well as I will discuss that what is the current research which is going on related to the zebrafish in neuropharmacology, as well as in some other areas like for the study of anti-cancer drugs, for the study of some hormones and some other relevant compounds. That mean we can say the efficacy of the various drugs using the zebrafish. So coming to this, first we know that what is zebrafish. So as you know that the name indicate that it is a fish which contains some stripes on the body of on the superficial layer that is similar like a zebra. So that means due to the presence of these stripes on the body of a small fish, we call it as a zebrafish. Now the question is that zebrafish how can be used, how can be maintained, how we can maintain some lab conditions for the experimental purpose, as well as for some other if we want to breed it, how we can maintain the zebrafish in our lab. So firstly I will discuss here that zebrafish is a small species that is having a length of nearly 1.5 to 2 inch, that is approximately 3 to 4 centimeter. Now this is a very small fish which is commonly found in rivers as well as ponds, but now it is also preferred as a experimental animal and experimental tool for the study of various drugs. Now the question is why zebrafish is preferred over the rodents, even though we are having some rats, some mice, rabbits, guinea pig, hamsters, various animals are there. Some are rodents, some are non-rodents. But if we want to prefer zebrafish as an experimental model, then what will be some of the critical things, what will be the importance of the zebrafish over the rodents, that I will also discuss here. So as I already discussed that this zebrafish is a small species with a very small size, with a body weight of approximately average body weight 500 mg. So which is too small that we cannot compare as with the rodents, comparable as compared to the body weights. But even though in some labs nowadays the zebrafish in our we are also using in our ISF College of Pharmacy, and we have also developed various animal models using zebrafish, like the animal models of Parkinson disease, animal models of Huntington disease, animal models of Alzheimer disease, as well as if I will discuss more about that, this is a good species for the toxicological studies. This is also a good species for the study of such type of drugs which are responsible, or such type of the agents which are useful for the regeneration. Because in case of zebrafish, the major importance is the zebrafish is having a good regeneration power. So if I will talk about the heart, in zebrafish, can regenerate his own heart within few seconds, that is 2 to 3 seconds. So that means this type of regeneration power which is retained in the zebrafish you cannot find anywhere in some other animals. So that's why the second thing, if I will discuss, the main focus of zebrafish nowadays is on the retina study. So that means the retinal pigments as well as retina regeneration power in case of zebrafish is too high. So that's why instead of that, the zebrafish contain more than 70 genes which resembles with the human. So that means if anybody want to study on some genetic models, that means zebrafish can be more suitable because the similarity index, genomic index similarity of zebrafish with human is nearly 70%. And if I will discuss that about some disease, in case of disease study, the average resemblance with the human is 84% around. So that means due to this, a large data which we can easily be taken from the zebrafish related to some genetic study. So that's why this zebrafish is more preferable animal. Now, if I will discuss that zebrafish species, they might be having a different colors like other species. Sometimes the outer appearance of zebrafish could be yellow color, blue color, black color. It depends upon the type of pigment which is present in the outermost cells of the zebrafish. So that's why zebrafish contains some kind of fluorescence. So due to that fluorescence, it emits different type of the colors. So coming to the next, that zebrafish, in which area we can use, because from 1960 the zebrafish is preferred as a good species, good animal for the study of various type of the drugs. So that's why after 1960 some of the labs they use zebrafish around in 1970 for the study of anti-cancer effect of some drugs. Afterward, around in 1980-88, the zebrafish was preferred for the regeneration studies. But instead of that, the zebrafish could be a good model in case of neuropharmacology. As I have discussed with you that within a short time we can easily develop as well as validate the animal model of zebrafish, because a zebrafish is having a short lifespan which is nearly 2 to 3 years, but this lifespan is quite near to the rodents. Likely in case of rat, as in case of mice, their lifespan is again 3 to 4 years. So the lifespan in term comparable with the rodent is almost similar. But if we want to validate or if we want to develop the model for by using the zebrafish, it can easily be developed. And secondly, the body size and the body weight of zebrafish is too low, so that's why it less amount of the drug, less amount of the toxin is required to induce any kind of disease. Instead of that, this zebrafish is also preferred in case of metabolic research to analyze the metabolic activity, because in case of rodents we know that mostly we prefer rat or sometimes we prefer mice for the metabolic studies, for the endocrine studies related to diabetes, diabetic neuropathy, and diabetic associated dementia. But in case of zebrafish, it is possible that if you want to develop animal model of zebrafish for the diabetes by using some of the toxins like STZ induced diabetes and alloxan induced diabetes and stress induced metabolic problem likely diabetes. So such type of the animal model can easily be developed in case of zebrafish, and anybody can easily analyze the efficacy and potency of the drug over these type of the species. Instead of that, the zebrafish is also preferred in case of immunology, because in case of immunology studies like immune associated studies, zebrafish is also widely preferred because zebrafish very fastly respond toward any kind of the antigens. Antigen can be environmental toxins, it can be some toxic chemicals, it can be some allergens. So that's why in some of the studies, in some of the immunological studies, the zebrafish from the last 20 to 25 year has been preferred. Instead of that, the zebrafish is used for the study of effect of gene and gene expression. So that's why in case of genetic manipulation, in say for genetic research, the zebrafish is also very widely preferred. And many of the genetic disorders which are associated, in which there is a link of some chromosomes, some genetic alterations are there. So that's why the zebrafish, which are very cheap as compared to other rodents, because if we see that zebrafish, before zebrafish there was a use of some butterflies, because as we know that we always initiate research from the small animal to we go for the large animal. So if the animal or the any the species which are we are using for the model validation, if the size is less and if it is small, then it is number of genes will also be less in number. So that's why it is very easy for the genetic studies to find any change at the particular target site. So after butterfly, the zebrafish is nowadays preferred for the genetic studies. And after then we can discuss that zebrafish is a good tool for the study of toxicities, because in case of rodents we can do acute, subacute, chronic and subchronic type of toxicity, but there are some limitations. The limitation mean we need a large number of animal for the toxicity study, and secondly we need a large quantity of drug that we are going to study. In case of zebrafish, they are very cheap and you can use a large number of species, and instead of that anybody can breed zebrafish in the lab, and large number of the zebrafish can be used as desired for the groups which they are using for the study protocol. So in case of previous studies which are reported, I have found, and even though we are using that n is equal to 12, 14, 18 like that, instead of rodents we prefer n, the number of animals per group, that will be around 8, 6 or 10. Likely we can calculate by using sample size calculation method, but in case of zebrafish we need a large number of species for the study of toxic effect of the drugs. After then that, it is preferred animal for the drug discovery purpose, because in case of toxicity on the rodents, we need that we must go through the firstly the toxicity of the compound, because it do not harm to the animal directly. So we need some in vitro analysis. Instead of in vitro analysis, the zebrafish could be preferred for the cytotoxicity studies. So cytotoxicity assays can be performed by using the zebrafish. Now coming to what are the physical facilities which are required for maintaining the zebrafish in the lab. So firstly, you know that as per CCSEA guidelines, previous name was CPCSEA, now which is the Committee for the Control and Supervision of Experiment on the Animal. They given some guideline that how we can maintain zebrafish in our animal house. The first thing they need that building corridor and utility. That building, that the walls must be properly sealed and there should be no any kind of leakage. They must be moisture proof, and there should be a proper ventilation of the air as well as light. So this is the first thing which is required everywhere. But in case of zebrafish, the little difference is there from the rodents. That in case of zebrafish, we need 14 hours day cycle and 10 hours night cycle. Instead, in rodents, there is a 12 hours light cycle as well as 12 hours day cycle. Now, the second important thing in zebrafish is that the zebrafish is a light resistant. So instead, fire resistance as well as light resistant. So that's why if you want to retain in zebrafish in any lab, you must be internally seal it with any kind of the sealing material, or either you can apply black color charts over there, so that zebrafish will not be directly explode toward the sunlight. The second thing is zebrafish, where there is a some fish, that mean how we can the door which will be there for the lab, that must be rust free, and that door must be properly moisture proof, and that must be clean, free from any kind of the dust. So there should be no any kind of the cracks, no any kind of the hole will be there, so that can be responsible for the contamination. Now the third important part is handling of the zebrafish. Now basically that there are so many techniques by which we can handle, because sometime in some studies we need to carry or analyze the body weight of zebrafish. For that purpose, what you can do, you can easily keep the zebrafish out for few seconds from the water side, and you can keep it over a wet cloth or any kind of the substance which previously must be wet with the water, so that zebrafish for few seconds can easily be maintained outside the water tank. So that's why handling is not difficult, but it needs some expertise and some skills. And the second thing which is required, that how we can administer a drug or a compound to the zebrafish. So basically that it is a critical thing, that if it is not easy to give intraperitoneal or oral solution to the zebrafish. But if anybody want to give a intraperitoneal solution, that range as per CCSE is 1 ml per 100 gram of the body weight. So the solution or the drug which we want to give by IP route to the zebrafish must be nearly 1 ml per 100 gram of the body weight. Now the second important part is, if you want to collect the blood from the zebrafish, because in some studies, that if anybody is doing a work on zebrafish, that is ST induced diabetes or some other, in case if they need to collect a blood from the zebrafish, it also requires some skills, and it also need that the scientist or the researcher whosoever want to take the blood must have knowledge that how much quantity of the blood can be taken at a time. So that's why the maximum volume of the blood from a zebrafish at a time can be taken, that is 10 to 12 microliter. But it must, if you want to take 2 to 3 times in a day, it should not be more than 1.2 to 1.3 microliter. So that's why average quantity of the solution which we administer either by IP route, that must be 5 microliter nearly, and average quantity of the blood which you can withdraw from the zebrafish should be around 2 to 5 microliter. So next, if we coming that how we can maintain the water quality of the zebrafish, because zebrafish they survive only in water, and the water must be in a tank, and the tank must be a transparent, so that you can easily analyze or check if there is any kind of dust particle or whatsoever the thing is there. So that's why it is important. And if you want to retain it, the water temperature must be around 26 to 29 degrees centigrade, and the optimal temperature of water for the zebrafish is around 28 degrees centigrade. So the water must be clean on the daily basis, and if you are unable to clean on the daily basis, it can be done on the alternative days. So It is also necessary that if you are finding that the water temperature is too low, then how you can improve? You must add some of the warm water very carefully without direct exposure toward the zebrafish. Then you can retain the optimal temperature. The next thing is pH. The pH of the solution or the liquid where you are retaining the zebrafish must be nearly 7 to 8. So it also need to maintain on the daily basis. If you found there that pH is too high, then you can reduce the pH by adding a few drops or a few quantity of sodium bicarbonate. And if you found there that the pH is too low, you can increase it by adding a dilute HCl. Now, next important part is conductivity. It is not necessary to maintain, but salinity is required. That how much the water is saline, because you know that these fishes are the pond fishes. They mostly live in the ponds and rivers, so that's why they need some quantity of salt. So we must add sodium chloride, that is nearly 5 gram per 1000 ml of the liquid, so that we can maintain the salinity of the solution. And then we also must check the hardness of the water. Now coming to next, alkalinity, how much the water must be alkaline. So it the normal range of alkalinity will be 50 to 75, and alkalinity of the solution must be analyzed on the weekly basis. So you know, if it is too alkaline, you can improve it by adding some of the RO and iodinized water. And then if you want to reduce it, you can add sodium bicarbonate. So the quantity which is required, it normally you can easily understand, or you can check at that time that how much the water is alkaline. From that you can easily understand what quantity of sodium chloride or the other salts that is required. Next is important part, which is a dissolved oxygen, because you know that we provide a aeration in the tank. The main aim of the aeration is to provide the oxygen to the zebrafish. Particularly for the normally, we maintain the aeration approximately 40 to 50 aerations per minute, and you can increase or decrease because it depends upon the quality of the instrument or the apparatus which you are using. But the aeration must be proper so that zebrafish can be live for a long time. Next important part is that there should be a no presence or no addition of chlorine in that water. So chlorine, there is a no need to add chlorine in that water, because if we add a chlorine, it reduce the body weight of the zebrafish, and it also interfere with the normal aeration oxygen consumption rate of the zebrafish. So that's why this is important or necessary to be checked. After then, there is a need to analyze some nitrogenous compound like gaseous. What will be the normal value of that nitrogenous compound, like ammonia? You know that in case of rodent, if we keep the rodent in the animal house, the ammonia level is increased. But again in the zebrafish, the its level must be less than zero point 0.5, and for the nitrate its level must be less than 50, and for the carbon dioxide it's nearly 15 parts per mole ppm. So these are the basic thing which we have to maintain. So this was about a zebrafish, how to retain and how to maintain in the lab. You are welcome. Thank you very much.
Yes, sir. Any issue, sir? Okay.
It's okay, sir. Thank you.
Sir, wait a second. And you can turn off the presentation. So let me.
Okay, thank you, sir.